Yes, and a good morning to you. It's good morning, Friday morning. Um, and it is, boy, flying by here. We just have, um, we're, we're looking at March already. To me, it's like barreling down on us. Yeah, and we, I've made a little adjustment to the exam due date, just an FYI. Um, so I originally had made it for the 20th and changed it to the 21st. Not going to do that every time, but the first exam I thought I would definitely do it. I'm also thinking along the lines of making it due on a day that we do not have class, so that just time um, management might be helpful. At least that's my intention. So it is due on Tuesday, um, Tuesday the 21st at 10 p.m. and not Monday the 20th at 10 p.m. All right. Um, and actually, oh boy, I completely botched that. I meant the completely botched that. Today is the 19th, so 20th, 21st, so the 22nd. I mean, the 22nd. Um, completely. Where the assignment is still due the 21st. The assignment is still due. The homework is still due in the same time period. I'm thinking along the lines of the exam and making it on a day that is it's not a day that we're meeting. So, are there any questions about that? Let me just say, I think I might have made a typo. I may have made a typo. Let me go in and look at the Dropbox. Okay, so the exam I have made due on the 23rd. That's a Tuesday. Um, and just make sure that we have a Dropbox still set up the way I had planned on it being. Dropbox is due on the 20th. Okay, so that is tomorrow. Um, and I will be, all my classes actually have assignments due this weekend, so I'm a little bit overwhelmed with grading, um, and definitely definitely have intention to getting in the within the next week your assignments graded. Um, Gabby asked if there's a good way to study. Well, um, if you go into each chapter, um, there are a couple different mechanisms to use to study, and these are associated with the textbooks resources. So these are all linked up, um, and yeah, my PowerPoints are here. Kind of tell people that you're, and also have exam one reviewed, by the way. So this is definitely for um, for that very purpose, Gabby. Um, and I made this quite a while back, but it's um, still very relevant. It's the entire unit's review, and it doesn't give you answers, but it gives you the subject matter of our discussion. Okay. Any other questions about the exam? If you look at the topics here that I have for each of the chapters are laid out, 1 through 4. Okay. Any other questions? All right. So let's get the golf ball rolling here. Um, and what we've been covering medallion traits so far. That's been our focus. So then we're going to go into um, polygenic traits, which where there is a way to do a Punnett square with polygenetic traits, but we're not going to worry about that. That gets into a you might have to do that in your biology course if you're go that direction with your science, but we're not going to do that here. But we will talk about the concept and what you would be doing in a Punnett square if you were so to try to do so. So what a polygenetic trait is, as opposed to a Mendelian trait, these make up most of our traits that we see, most of our phenotypic traits that we see. And they are a combination of multiple sets of alleles. So it's not like a Mendelian trait where you have one set of allele that is kind of two sides of the same coin. You have multiple coins that are influencing a particular phenotypic trait. These traits are expressed on a continuum. We'll more on that in a minute. They are measurable. They are, like I said, they comprise majority of the traits that are really very prevalent in our minds when we look at other people. These are obviously observable traits, as all phenotypes are in some way observable. So what are some examples of this? Anybody have any immediate examples? What about what is it that we see the most of folks? What is it we have the most awareness of each other when we look at each other?
Are you referring to like the actual visual? Like if you look at the other person, you can distinguish characteristics.
Yeah, exactly.
Oh,
Exactly.
okay. Well, I
Most
guess
all of
like
our,
eye color.
yeah, exactly. It's what we judge folks are, what we sort of, how we calculate what group they belong to, you could say, how we racially categorize folks. And we're going to break down that category of race later on the semester. But, we're talking about eye color, our skin color, our hair. This is, basically it is, it's everything on the surface. So like I said, these traits you can measure. So you can measure your height, your stature. You can measure how tall you are. And everything can be calculated within a range, a given range, versus just, so what we're talking about here is the difference between continuous distribution of a trait versus discrete. Discrete data is you either have it or you don't, like in the case of ABO blood typing, and typical Mendelian trait in which you flip a coin, you get a head or a tail, and you can represent that it's very simply in a bar graph. When you are measuring the height distribution, you are you are measuring something obviously between however many feet versus how many feet, and that is when you are we'll talk about this later in the semester, but if you are to take a walk from say Africa to Scandinavia, long interesting walk I would say. It would be, and you would see the traits change as you walk through that path. You would see a continuous change, and as we'll talk about, it is in that change typically is in response to the environment, and particularly if you look at skin color. It's a continuous change of distribution of skin color. Skin color is a little bit more difficult to calculate, to necessarily measure, but there is a way to do that. So yes, you could with discrete data, the difference between calculating the number of students in a classroom versus the continuous data that would measure the height range of a classroom, or the weight of somebody in a classroom. So yeah, continuous versus discrete. This is a kind of a signature of when we talk about polygentic characteristics. Skin color is one that we fixate on because it's so in our face, and it's has such a powerful influence on how we see people. At least it has historically been very used as a power source, right? So in this class, we like to break down race and say it doesn't exist, but it does exist on a social level, but it doesn't exist biologically speaking, because it's such a superficial, as far as significance go, as to who somebody is biologically speaking. It is. I believe I've read that somewhere between 6 or 7 sets of alleles that are very influential to the pigment tone of your skin, and of course, the pigmentation of your skin is defined by the amount of melanin production that is in direct response to the sun. Again, we'll get into a little bit more of the weeds of this in, I think, chapter 12. But you know, this is again, it is environmental factors that are really important here, and this is in all polygentic characteristics. Environmental factors are important, and so what we're looking at, for example, in one's height, we can see historically that humans have gotten taller and taller as we have evolved over time from our environment. And so what has happened is the diet has changed, to say, for example, and not only diet whenever somebody is living, but the diet of the mother before birth. So whatever the mother does maternally influences the development of the fetus. So that always has an environmental element to it. Again, when we talk about sunlight and the exposure to sunlight, we need a balance of sunlight to be a healthy human being, and we will develop deficiencies that will lead to potential health issues. Again, we'll get a little bit more into that when we talk about skin in chapter 12. So this is also we have toxins now that we didn't have back 100 years, 200 years ago in our environment. Our exposure to toxins can influence our phenotype, and therefore can influence our genotype. Our genotype, our genes and our phenotypes are always doing a dance with each other based upon our environmental influences. But the genotype itself and the genetic factors that we're going to look at here in a minute are definitely still very important in this equation of what the phenotypes become. So let's look at the breakdown differences between Mendelian versus polygentic traits. So Mendelian traits, remember, you have only one gene, one set of one address code, one location, one locus. Versus polygentic, you have multiple locations, multiple genes that are influencing. Mendelian traits, environment is not important, but polygentic traits, they are. Okay. In the distribution of the phenotype of the Mendelian traits, it is a discrete, you either have it or you don't. And with the polygentic, it is a continuous, non-discrete distribution. So are we clear on the difference between polygentic and Mendelian traits? Are there any questions of concern? Okay, silence is scary, but it's also golden with the anticipation that y'all know what's going on. So
I'll break the silence. It sounds good.
Okay. Great, thanks. All right. So two definitions of evolution. We have been talking about Darwinian definition of evolution, which emphasizes this deep time that we've talked about, that evolutionary traits evolve because of deep time and puts an emphasis on natural selection, right? So populations change based upon natural selection. And with that is the backdrop to what we've learned in the 20th century. We have progressed into what we call the modern stage of understanding of evolution, which is looking at the genetic allele frequency of a population. And the modern understanding still recognizes the importance of the changes in populations over deep time through natural selection. However, there is evolution always happening in the micro level. So the micro is the genetic world where you're having these these alleles that are potentially mutating in meiosis. Or in other ways from various sources, which we're going to look at. But meiosis is what we've covered so far. But there's also things happening on the macro level as well with natural selection. So the modern evolutionary theory, so there was this debate all the way up till about early 1900s about what was happening there. And remember, Darwin did not have a window into the genetic domain. So, but the debate is up till I'd say 1930 or so, 1920, I think, was between gene mutation or natural selection, which was the most important factor of the internal world or the natural selection world that can be so observed so well. So what was agreed upon was that allele frequencies are changing, and there are many ways that these allele frequencies in a population are changing. So when we talk about populations, we're going to define population here in a minute, but population, a group of people, group of individuals in a species that are mixing together their gene pool, right? They're interchanging their genes. In that population, the frequency of sets of alleles which are coding for a phenotype, they're changing, they're morphing. So this modern synthesis recognizes that there is production of genetic variation in a population that is happening on three different levels. Talked about gene mutation at the DNA level. We've talked about meiosis, shuffling of the genes, the sort of randomness of the genes, which also you find mutations existing there, of course. You find mutations happening. We talked about the protein synthesis. So those are the two things that we've recognized so far. But there's a third element that we haven't talked about. Does anybody know what that third element is that is producing genetic variation?
Pressures.
Okay, yeah,
From
pressure
the environment.
from the environment. So that is associated with natural selection, right? So that typically has always thought about deep time, but it's been shown actually to happen kind of pretty quickly, actually, because environment is changing. And it's we're looking at Texas right now where we have this extreme change of environment. Boy, there's some species in Texas that are just being, particularly humans that we know of, been thrown for an environmental loop. How do we respond to this pressure? But I just came to my mind that, well, this is evolution happening right before our eyes with what's happening in Texas right now, where they never get this kind of extreme weather. Even our culture is having a hard time adapting and is not adequately adapting to the changes that are happening in Texas right now. So yeah, but when we talk about genetic variation, the three levels we're talking about here, genes. So selection is going to pressure genes, because when we talk about environmental pressures, it does select individuals who can survive certain environmental changes. So therefore, there is some who can reproduce successfully will be coming out of that. But the third, what we're interested in, is gene flow, which is associated with environmental pressures also, as I'll explain. But gene flow, and that is when populations are there's multiple things that could be happening here, but there's typically a reduction in genetic variation that takes place. And so we don't like reductions in genetic variation. That means some people are dying off, that they're not surviving, or some individuals aren't surviving in a species. So reduction of genetic variation is something that happens through a couple different vehicles. One thing that we have to note is that it's necessary for this genetic variation to exist, for populations to continue to exist and to evolve. You've got to have a change in the allele frequency. So individuals who die as a result of this, it's not something that, obviously, as an individual myself, I like how things are going. I'm glad I had kids. I'm glad that things worked out. But for a population to evolve, things have got to change inside of the gene pool. So the question is, what is a population? And like I mentioned already, if it's a population that is interbreeding with each other, right? So the human species has been reproducing itself, interbreeding with itself. There has never been long enough periods of time for the human species to isolate itself into an island, such that when I say the human species, the modern day Homo sapien, which has a couple different calculations as to how old that modern day Homo sapien is, but let's go with 100,000 years right now, but we'll talk about the debate between that later in the semester. But in the 100,000 years that modern day Homo sapien, who migrated out of Africa and populated the world, there's been no long enough stretch where a group has isolated itself genetically to become and evolve into a species separate. Species are define a population that can interbreed with itself. Any questions about this? Okay, so there is a way to, like I said, populations have to evolve, have to change to continue to respond to environmental change and to continue to basically survive in order to adapt. So there is a way to calculate. The book talks about this a little bit more in detail. I'm not interested in you to actually understanding how you calculate the stability of a population, but I want you to understand the concept behind it. So if we take in a hypothetical set of alleles, and this is Mendelian trait, hypothetically, if we have the dominant capital B and the recessive lowercase b as being a hypothetical trait, you have three different combinations of those alleles that are possible. Capital B, lowercase b, or two lowercase. So we got two recessive, one dominant, one recessive, or two dominants. You add up in that population that has that particular set of alleles, you add up the number of people based upon those that possible combination, those three possible combinations, you'll be adding up the entire population. That's 100%. So what can offset that stability? This is the snapshot when you're judging, when you're trying to test how stable a population is. You'll see first off from point A how frequent these three combinations exist in a current population. Then 5 years from now, you will test that trait again to see if those three combinations have changed. So there's been some offset to the stability of those three genes. What has created the offset? What has created an instability in one of the three? Well, gene flow, and we're going to look at each of these a little bit closer. And this is gene flow is any change in the gene pool from one generation to the next. And the gene pool from a population breeding together shared by, so each baby draws upon the pool of population. So everybody that's born, individuals that are born, they are drawing upon what they have inherited from their genotype, right? So their parents' genotype, and their parents have drawn from their parents' genotype. So the passing down of those alleles from generation to generation to generation, and of course, like we talked about, mutations happen, change happens to those genes. So there's also what we call genetic drift taking place, and these are very much again, this world of randomness that takes place. Again, we'll come back to these individually. Then there's what we call bottleneck events, and individuals are lost out because of bottleneck events, and there's a reduction in the genetic variation as a result. So let's start with talking about genetic drift. Gene flow is something that we've somewhat already covered, but genetic drift is something new to the equation and to our discussion. Genetic drift, like I said, it's random. It is if there's a major catastrophe. So in Texas, I don't know how many individuals have died so far, but let's actually take a different example. Let's look at let's look at the coronavirus. So I believe we've had 400,000 people die, if I'm not mistaken. If that's where we are now, it's slowed down, but in a population of 350 million, losing 500,000, 400,000 is somewhat significant. The question is, those people who died, are they taking with them a set of genetic alleles, sets of alleles that influences the people who are remaining? So if you take a little bit more of a smaller population example, the Inuit, who have been in bad shape as far as their numbers for quite a while, like a lot of Native American groups. If you have an Inuit group that has only 150 people in it, and 10 people die, 15 people die, 15 people die as a result of the virus, that's a significant proportion of their population. And so their genetic pool will be impacted. This is genetic drift in action. Any questions? Okay, so a third possibility, thank you, Stephen. A third possibility is the founder effect. The founder effect is it's also referred to as a bottleneck. And you have, typically it happens when migrations take place in human populations. So When a group migrates to um, a smaller group of a larger group migrates, and this is more relevant to earlier times of human population growth, but when humans were not spread out around the world as they are now, when a small group would leave and set up camp in another part of the world, they would take with them a sample of their alleles to another part and set up. And whatever that sample is, it will definitely have a reduction in diversity of what they had before. And so whatever they have, they're carrying with them is going to populate and explode into what is definitely more defining of their new settlement. So you have an image here that sort of well demonstrates, illustrates what we're talking about. The original population has a multiplicity of colors there that include a pretty predominant red, as what I see is kind of the most standing out color. But the population that left camp and went somewhere else, they did not have many reds. So as you see, they have a prevalence of yellow and green. So loss of diversity took place. Some examples of this. An elephant seal, which was all over the place before humans, like a lot of large species, overhunted. And we'll talk about elephant seal because these are darlings for physical anthropology for us to use. The male is about 5,000 pounds, and the females tend to be about 1,000 pounds. So males dominate the females for sexual selection. We'll talk about sexual selection in the next unit. But in the hunting game, they were nearly wiped off the face of the earth. There were like 20 individuals left in the 19th century, at the end of the 19th century. They have since, because of being considered to be an endangered species, have rebounded. But the individuals that survive, those 20 individuals that survive, there is a definitely bottleneck that took place. And so there's much less genetic variation. And that's not good. That's not good at all, because when you reduce the variation, then you are reducing the ability to respond to environmental pressures. And yeah, Rebecca, I do think that I like I said, in smaller populations where COVID took a more devastating influence, that's a good question. I think that the science is out on that. And I don't think we know for certain how COVID is going to influence the overall genetic pool of human population. Millions of people have died across the world. The virus targets obviously vulnerable certain populations, typically associated with problems that are lead to early health issues already, whether it's high blood pressure, definitely things that actually aren't going to prevent from reproducing, because most of the problems that made you susceptible to the COVID virus would not prevent you from reproducing. So we'll talk about that in a minute, but it's a good question. It's I'm not for certain, to be honest with you. I'm not for certain. Another example of bottleneck would be the settlers who, in the 17th century, went to South Africa. One of the first colonists that came from Holland. We're talking about the Dutch. These Dutch settlers went to South Africa, and it was just like, I believe, a couple of hundred colonists, couple hundred folks who settled there, and but they brought with them, and these were all pretty much all men, and they brought with them a set of alleles that were horribly tainted that led to Huntington's disease. So South Africa has one of the highest percentages of Huntington's disease, which basically is it's a death curse after age 40, and there's not much surviving. So that's a great example of a bottleneck. Another example of a bottleneck would be looking at the population. We know that the first population that migrated from northern Asia down into the Americas happened about 13,000 years ago. And we know also that that population was a small group, and they had a definite reduction in their diversity that they carried with them. And there's thought that the reason we have, as far as the ABO blood typing group, there is 0 Native Americans as far as the B frequency. And so it's thought that the Northern Asians who came over and became the Native Americans and populated all of North and South America had very little as far as B blood type. So yeah, this is the map to remind you that there is nothing in the Americas that indicate the B allele. So there was something that took place there with that migration. It was a bottlenecking effect. Any questions about bottleneck? Right. So this brings us to the question of natural selection and how it acts on our phenotypes and how it creates variation, how the pressures of our environment produces variation. And there is a thought that considers the natural selection to have a goal. And what we really want to look at is it's not goal driven, but there is a process that has direction. And if you go back to our finches on Darwin's Galapagos Island experience, the size of the beak was driven by the weather on each island and the food available on each island. So the size of the beak changed as the weather changed the food that they were eating. And so you need a different size beak to eat different types of food sources. And so this change in beak size, it has a direction. And if there is a major change in the weather that from one year to the next, like we're seeing today, that is becoming drier, then the beak size, you need a more robust beak that is going to be able to eat the seeds that now have become hardier to eat because of the dry climate. So how that influences the genetic distribution of alleles is going to be based upon 2 different factors, mortality and fertility. So, and that's what I want to talk about next. Mortality is, do you live long enough, or if you die before you can reproduce, therefore you're not going to pass down your gene pool. So differential mortality is driven by those numbers. If you don't live long enough to reproduce, then your gene pool is going to be eliminated from the equation of future generations, right? So fertility, on the other hand, deals with those that can survive and can pass down their genes, their alleles, and they can survive that bottleneck. And so therefore it is your allele pool is being differentiated based upon the people who can be fertile and reproduce the next generation. So those are the 2 elements. A lot of diseases influence, you know, like cancer. A lot of the cancers, you know, are happening late in life, so they've already been passed down. Cancer is already, the gene equation for cancer is already been passed down. So to highlight all of this, we need to look at sickle cell real quick. Again, sickle cell, just to remind ourselves what we're talking about here. Sickle cell anemia happens in tropical regions of the world, Africa, Mediterranean regions. And it is based upon one set of alleles, one gene. And that gene, like the AB O blood typing, is codominant. It is codominant. So you have A, which is normal hemoglobin, and S, which is the sickle trait that is problematic. Yes, Nancy, good example. Color blindness is definitely something that is a great example of the bottleneck. I think the bottlenecks are, the number of examples are infinite, actually, because they're always happening. And this is a great example of a bottleneck too, is the sickle cell, because what's taking place here is that if you have normal, and what's the main pressure point of the sickle cell is well, there's two pressure points. One is malaria, and the other is the trait of S, the sickle trait, the sickle allele, because normal hemoglobin, which is AA, that's what most of us in this part of the hemisphere, the North America, South America world, and most of Europe, north of the Mediterranean region, most of us have this normal hemoglobin, which is sickle cell. So normal hemoglobin is circular. Sickle is shaped like a sickle, and it doesn't flow through your veins smoothly. So it gets clogged, and people die pretty young as a result most of the time. So in the area where you have malaria, you have the what has evolved is this sickle trait, because if you have 1S, then it provides a resistance to malaria, and so that is a good thing. So if you have 2S's, though, that's the early death sentence. So you got two different pressures. First off is malaria, that will kill somebody with normal hemoglobin, an AA person, but will not kill somebody that's an AS person. And if you have a parent, if your parents are both survivors because they both have AS is codominant, they both are being expressed, then your parents will have a 25% of having kids that are with the sickle cell disease. So it's kind of a catch-22. The AS allows for individuals to survive, but if they marry other ASs that survive because they have AS, then you got a 25% chance to have children that have the sickle disease. In the Mediterranean region, this was all sort of diagnosed by looking at overlaying the maps of the sickle cell trait, that is the killer trait of the sickle cell disease, and the malaria distribution. And you see there's an overlap. And so this kind of scientists were able to sort of piece this together. And so, just to break down this history, the S allele there's no real advantage to having the S allele in North America. So it evolved in this region, it mutated, became important in this region to survive malaria. And malaria is a product of the farming. So Africa regions of the world did not have farming for a long time until farming evolved and human populations. And so when farming developed, they clear-cut trees, and pools of water started to develop as a result, and mosquitoes are attracted to those pools of water, and that's of course, mosquitoes are the spreading of malaria. The S allele frequency increased because S protects against malaria. Number 4, the frequency of S is as high as 40% in some populations as a result. And it's more frequently, of course, present where malaria exists. So that's kind of the dynamic. The question is, what's happening here? Is there a goal with this natural selection? Because we're selecting for people with the S allele, right? And that's something that it's a process. It's not necessarily doesn't have a goal to wipe out the people who are all AA. It's kind of a random process that the evolution is acting in a certain direction. So yes, I wouldn't. There is a goal. And there is a process that is a direction, has a direction, but it is not goal oriented. And when we talk about fitness and our ability to survive in certain environments, this is something that is, it is by luck and by chance. Having an AS in the Mediterranean region is an advantage. But it's also a detriment as well. It's you marry another person with AS, it could cause a problem with your children. But if you had an AS in North America, there is no advantage at all. So it is definitely what is normal. Hemoglobin depends upon where you're living, and it's all about the context. It's all about just random luck. And so what we would like as far as our ability to adapt to the environment, the more diversity you have in your gene pool, the better off you are. But also to strategies to survive. We're going to talk about this more about in a different chapter, but just to give you a little bit of a, we'll talk about this in macro evolution as well. But species that are more generalized, that is, they have more mechanisms for food sources. They don't rely upon one source of food. They have more ability to adapt to their environment. Specialized species that respond to only or rely upon only one source of food, that makes their species more vulnerable, to say the least. So specialized versus generalized species, again, we're going to talk more about this in chapter 5. But a great example of this is koala bears, which are not really a bear. They're a marsupial. But it's a nice example because in comparison to the black bear that eats everything, black bear is in much better shape, and they can adapt to their environment and environmental influences because of that. Koala bears, on the other hand, they had a hard time during if you remember hearing about when the fires were taking place over in Australia, their food source was being destroyed by the fires. So they were at a high risk as a species during the fires. So yeah, I mean, this is somewhat covers, again, it was sad. It was, they had, you looked online, you could see pictures of people saving the koala bears from the fires. So this has covered the full range of evolutionary influences that we want to cover for this chapter. Are there any final questions? Okay. You can email me any questions you have between now and Monday if you're taking the exam over the weekend. I will be doing some grading on Saturday and probably some on Sunday as well. Okay, so Stephen, if you have some questions about the homework, you can, if you want to hang out after class right now, I'll be glad to talk to you. If you want to email me any questions you have, then that's good as well. So great. Besides that, everybody have a good rest of your day. And Monday we'll be starting chapter 5. We do have class on Monday. So look forward to getting into the world of macro. Have a good weekend. Thank you. Thank you.
The only question I really had was you asked about in the homework the probable genotypes of a non-tongue roller.
Yes.
Did you want the probability or the actual genotypes? This is probably a dumb question, but I was having some trouble thinking that one out.
They all make you think each of them make you really think through some possibilities. I believe that question is asking for the percentage. I have to look at it myself to
I could copy paste it in the chat if you would like, but
do you have it right there?
I do, yeah.
Okay, go ahead. Yeah, so let me look at the larger problems. That's part of a series of questions, and I have to kind of look at the context of the question. Let me grab that real quick.
Gotcha.
I can open that up really quick.
I hope I'm not holding you over too much. I'm sorry about that.
No, you're good. You're good. No, this is I definitely I always have time after class. Well, I think of that. Sometimes I have a 10 o'clock appointment, but I'm good today. Let's see what we got here. Okay, open up Sesame. Okay, so ability to roll the tongue. The ability to roll the tongue in a 2U shape is an inherited dominant. The trait expressed with an R, while the inability to roll is an inherited recessive trait. Write the phenotype of the following. So you can write the phenotype there for each of these. So what is the probability of genotypes of a tongue roller? So if you have a Punnett square, what is the percentage? What are the potential combinations of genotype? So yes, and for both of these, how would you? What would be the genotype of somebody that has a phenotype of a tongue roller?
I am glad I asked.
Yeah.
Yeah, that would have been something else.
Same thing with a non-tongue roller as well. So genotype is defined by your set of alleles that lead to a phenotype.
Yeah, much appreciated. I appreciate the explanation, man.
Sure thing. No problem.
Have a good day. I hope you have a great weekend.
Thanks. Have a good one yourself.
Adios.
Bye-bye.